Hi, my name is Bhavik Patel, and I'm going to be talking today about experimental hydrocephalus. I have no relevant disclosures. Hydrocephalus research is a rapidly evolving area of research, and there are many questions that remain to be answered regarding hydrocephalus. I've listed a number of questions here that remain to be investigated, and you can see that it ranges from understanding CSF flow dynamics and what the causes of fluid dysregulation are in various types of hydrocephalus, all the way to understanding genetic mechanisms responsible for hereditary hydrocephalus and how epigenetic changes may occur in the setting of hydrocephalus, and all the way to understanding different ways in which we treat hydrocephalus and finding new and more effective ways of treating hydrocephalus. I won't read all of the questions listed here, but this full listing is in our text as well. In order to study hydrocephalus, several animal models have been developed and continue to be developed. A commonly used animal model is the injection of kaolin into the ventricular system. This causes an obstruction of CSF flow by mechanical blockage and focal arachnoiditis. And the advantage of this approach is that the timing is precise. A greater number of animal subjects go on to develop hydrocephalus than compared to other methods. And both obstructive and communicating hydrocephalus can be modeled depending on the site of injection. The disadvantages of this model include that the hydrocephalus tends to be rapid onset, which may not model what happens in humans, and kaolin may cause global inflammation. Potential subjects for this type of model include mice, rats, ferrets, piglets, dogs, and sheep. An alternative to kaolin injection is the injection of blood into the intraventricular system. The advantage of this approach is that it more closely models what happens in humans and therefore may be more clinically relevant. The disadvantages of this model are that the yield of animal subjects that go on to develop hydrocephalus is lower and is more variable, and multiple injections may be needed in order to induce hydrocephalus. Alternatives to kaolin and blood include virus, bacteria, and neurotoxin injection into the intraventricular system. These are older methods that are not widely used at present and have the disadvantage of the foreign substance being injected causing multiple pathogenic effects that make studying hydrocephalus more difficult in those animal models. There are also animal models for CSF shunting, which is the predominant way in which we treat hydrocephalus in humans. Sheep, pigs, dogs, cats can all accommodate commercially available shunts and can more closely model the systems that we use in humans. Alternatively, customized experimental shunts can be developed for mice, rats, rabbits, ferrets, and other smaller animals. Interestingly, animals develop shunt malfunctions at approximately the same rate as humans, and so this is a rich area of study. Are also being used very widely. The most common model is the rat hydrocephalus Texas model. And this is a model of congenital hydrocephalus that results from aqueductal stenosis. And these animal subjects will develop hydrocephalus within days of birth and can be treated with shunting. You can see how this is somewhat similar to what might happen in human newborns. There are other animal strains or other inbred strains of rats and mice, and the main disadvantage of these models is that the size limits CSF shunting experiments. In addition to that, they often develop severe ventriculomegaly very rapidly, which results in a limited lifespan. And congenital hydrocephalus represents a relatively small subset of the hydrocephalus phenotypes that are of interest to researchers. In addition to the previously mentioned, transgenic models have also been developed and are used to model hydrocephalus through a variety of mechanisms, as I've listed below here. For example, ciliopathies, which result in hydrocephalus, have been modeled in HY3 mice. Additionally, ependymal and subventricular zone alterations, which also result in hydrocephalus, have been modeled in HYH mice. Interestingly, Newer methods of creating transgenic models, such as utilization of the CRISPR-Cas9 system, have already been used to create animal models for hydrocephalus, such as in the L1-CAM rats, which have adhesion molecule and aquaporin channel impairments, which then result in hydrocephalus. As more and more techniques are developed for developing transgenic models, more transgenic models will be created. And you can see how transgenic models allow for the study of very focused mechanisms of the development of hydrocephalus. In addition to the in vivo modeling systems for the study of hydrocephalus that were previously discussed, biomechanical and in vitro modeling systems have also been developed. Biomechanical modeling involves the use of mathematical models of biomechanical mechanisms associated with hydrocephalus. We know from CSF infusion tests in animals that the global intracranial compliance decreases in the hydrocephalic brain, and biomechanical modeling can help us study this further. It's valuable for formulating testable hypotheses, but is limited by the fact that the true material properties of the hydrocephalic brain are not well known. In addition to these types of models, in vitro models can be used and have been used to study cell adhesion and growth on catheters, or responses to compression, stretch, hypoxia, and pulsatility. Lastly, the Harris et al. Bioreactor has been used to demonstrate the immediate deposition of protein biofilms that form a substrate for macrophage and glial cell attachment, and these provided evidence for cell surface modifications that reduce cell adhesion on silicone catheters. Lastly, I just want to touch upon the magnetic resonance imaging methods that have been used to study hydrocephalus in animal models and in humans. Periventricular white matter injury, which is seen as periventricular T2 hyperintensities on MRI, has been extensively studied both clinically and experimentally using MRI. And the findings in hydrocephalus may represent transependymal flow of CSF or may represent chronic periventricular white matter injury due to periventricular ischemia that's caused by the compressive effects of ventriculomegaly. In addition to T2-weighted MRI, diffusion-weighted MRI sequences have also been used, for example, diffusion tensor imaging, to study hydrocephalus. DTI can be used to visualize periventricular white matter tracts in vivo and can be used to demonstrate the compression characteristics of hydrocephalus. DTI measures of periventricular white matter integrity have been correlated with outcomes both clinically and experimentally. However, DTI is unable to differentiate between inflammation, edema, and axonal loss. DBSI is a more recently introduced magnetic resonance imaging method for the study of hydrocephalus, and this can be used to resolve crossing fibers, injured axons, and demyelination. And preliminary use in a ferret model with DBSI has been very promising. With that, I'd like to conclude this brief discussion of experimental hydrocephalus, and thank you for your attention. I'd also like to thank my collaborators both here at WashU and MUSC. Thank you.